We are going to start polysaccharides. Polysaccharides are carbohydrates which contain many monosaccharide units. Poly means how many? Poly means so many. And as per this definition, polysaccharides are sugar polymers containing 20 or more monosaccharide units. Actually in the biological situation, they contain several hundreds of thousands of monomers. Examples are, cellulose. Some of them are actually linear chains. Cellulose is a linear chain. While starch, glycogen, etc. are highly branched. And glycogen and cellulose and starch consist of repeating units of glucose. Then how do they differ? They differ from each other in the type of glycosidic linkage. Of course, they have different properties and functions. Let us come to the structure of polysaccharides in detail. Basically, the polysaccharides can be divided into two categories. One is the homopolysaccharide, the other is the heteropolysaccharide. What is meant by the term homo? Means similar. See, the homopolysaccharides are made up of only one type of monomeric unit. For example, starch. Starch is made up of alpha D glucose units. At the same time, cellulose is made up of beta D glucose units. Starch is made up of, glycogen is made up of alpha D glucose units. So homopolysaccharides, they may be unbranched or they may be branched, but they are made up of a single type of monomeric unit. And what about heteropolysaccharides? You see, this red colored one is a type of monomer, the blue colored one is a different type of monomer. So a heteropolysaccharide is made up of more than one type of monomeric unit. It's made up of different types of monomeric units, and it may be sometimes in a straight chain unbranched, sometimes they may be branched. And we will go to each of them individually. What you see here in this diagram is starch granules. You know that this is a chloroplast. What you see here in this picture, in the first picture here, is a plant cell. It consists of chloroplast. In the chloroplast we have starch granules. The starch is made by means of photosynthesis. The starch is made up of alpha D glucose residues. It is a storage form of energy in plants. And in figure B, what you have is actually a hepatocyte. That means liver cell. In the liver cell, we have glycogen. Glycogen also is made up of alpha D glucose. Starch is highly branched. Glycogen also is highly branched. Then how do they differ? Actually, glycogen has more points of branching compared with the starch. And many authors say glycogen is the animal starch. Here you see the structural details of starch. Starch consists of two types of arrangements. One is called unbranched straight chain of glucose. We call it amylose. In amylose, there is alpha 1,4 glycosidic linkage only. The individual sugars are joined by means of alpha 1,4. Carbon number 1 of a sugar is bonded to carbon number 4 of the next sugar by means of a glycosidic bond. All the sugars are alpha sugars, so we say alpha 1,4 glycosidic bond. And remember, only the anomeric carbon is the reducing carbon. So wherever is carbon number free, carbon number 1 free, not engaged in glycosidic bond formation, that end is the reducing end, as you see here. Any other free end of the molecule is actually non-reducing end. So here you can see only the right hand side, right end of the molecule is the reducing end. And here you can see amylopectin. What's the importance of amylopectin? It is branched. What type of branching? Alpha 1,6 glycosidic linkage. Carbon number 1 of a sugar is bonded with carbon number 6 of another sugar, forming a branching alpha 1,6 glycosidic linkage. You see here this main chain has a long chain of sugars extended both towards the left and towards the right, while this chain branch chain that has no more sugars towards the right. Why? Carbon number 1 anomeric carbon of this sugar is already engaged in glycosidic bond formation, while it is extended by many sugars towards the left. Ultimately, how does a starch molecule look like? These are all the points of branch, and these are the individual chains of carbohydrates, amylose, and at the extreme right side is the reducing end. Remember, only one reducing end is there because the carbon number 1 of this sugar is engaged in a glycosidic bond formation with the carbon number 6 here, like that. What about the branching of amylopectin? As we found earlier, it is alpha 1,6 linkage. This is a branch point. This is a branch point. And whatever I told you just now is there. Amylose and amylopectin. And the glycogen is similar to this starch, except that there are more points of branching with the glycogen. And glycogen is found only in animal tissues. See here, this is the starch chain. Actually, since the starch molecule is formed of alpha anomer of glucose, this chain can fold into a helical structure. This chain can fold into a helical structure. Can you see something like a spring? And this helical structure can trap iodine molecule in those helices. If you are adding iodine into starch, these iodine molecules are trapped inside the helices of the starch. And you know, iodine starch complex is blue-black in color. In the laboratory, when we are doing an experiment for starch, actually we add a drop of iodine solution into starch solution, and immediately that mixture turns bluish black, and we say blue or black. And that is actually an experiment for the identification of starch. And here you see two molecules of iodine. There are two molecules of iodine in one turn actually. That one turn of starch consists of 6 residues of glucose molecules. This is amylopectin. This is glycogen. You find out and you just you see where there is more branching. Is it in amylopectin or is it in glycogen? And this is actually cellulose. Cellulose also is formed of glucose, but the difference is that cellulose is formed of beta D glucose, not alpha D glucose. Look here, this is a beta sugar. OH group is upward. For all the sugars, carbon number 1 has the OH group upwards, while carbon number 4 has the OH group downward. So there is the bond formation like this. This is a beta 1,4 glycosidic bond, and it is completely unbranched. That's the difference between starch and cellulose. Starch is made up of alpha D glucose, while cellulose is made up of beta D glucose. Point 2. Starch is branched molecule, while cellulose is unbranched molecule. And if you consider any two of the nearby sugar units, we say it makes up what we call cellobiose. Two beta D glucose molecules joined together by means of beta 1,4 linkage. This is making up a cellobiose. This is making up another cellobiose, like that. You might have already seen two alpha D glucose molecules when joined by means of alpha 1,4 glycosidic bond make up maltose. Here Beta D glucose molecules when joined together make up cellobiose. And this is a cellulose, a part of the cell wall where cellulose is present. A cell wall, plant cell wall made up of cellulose. You see, here is a chain of cellulose, another chain of cellulose, third chain, fourth chain, like that. The cellulosic cell wall is having that oxygen OH group projecting. OH group of one chain can form hydrogen bond with the oxygen of the other chain. So this hydrogen bonds between adjacent parallel cellulose fibrils are stabilizing these cellulosic structures highly.